again, and we can go to the next presenter, and that is Tran from University on Montpellier in France. Please.
Yes, I will share my screen. I think I have a problem. Do you see my screen?
Yes, it's perfect.
I think I've lost. I think I lost my screen.
We can see your desktop, but no presentation is running. But we can see desktop of your PC. Yes, now we can see presentation.
Okay. So hello everyone. My name is Anna, and I'm currently a PhD student at the International Immunogenetics Information System, and I'm working on in silico characterization of the gene repertoires of immunoglobulins and T-cell receptors of the various inbred laboratory strain of musculus. And the adaptive system defines multicellular organisms from pathogens, which are recognized by antigen receptors, which are immunoglobulins, also called Ig, and T-cells receptor, also called TR. And the antigen receptors genes are organized in several loci. So here we have the example for the human. The loci are localized on chromosome 2, 7, 14, and 22. And there is AGH, AGT, AGL for Ig, and TRA, TRB, TRG, and TRD for TR. Those loci comprise different gene types, so variable genes, diversity genes, joining genes, and constant genes. And the adaptive immune response is characterized by an extreme diversity of the specific antigen receptors. So there are 10 exponent 12 different Ig and 10 exponent 12 TR per individual in humans. And the synthesis of Ig and TR proteins requires DNA rearrangement, following by the splicing at the RNA level, and complex molecular mechanisms such as end diversity and somatic hypermutation that occurs in B cells and T cells are also at the origin of that huge diversity. So for this study, we decided to get interested in inbred mice because it's the most used animal model, and two strains are widely used in biomedical research. So we have C57BL6J, which is studied for the induction of obesity, diabetes, and influence of age. And we also have BALB-C, which is used for development of hybridomas and production of monoclonal antibody. All the strains are far from being fully and precisely sequenced and characterized for Ig and TR loci. So knowing and understanding the organization of these repertoires in the different strains is therefore essential for understanding the reaction of the adaptive immune system and for the choice of marine models in biology. So as I said in the title, the goals of my PhD subject is to characterize the genomic repertoire of Ig and TR in loci mice, TR loci in mice strain. So the first step is to annotate the loci. Then we'll do a gene and alleles inventories. Then we'll determine the feature for comparison of strain. And then we will describe allelic polymorphism, structural variation, and copy number variation. And for that, we'll develop a methodology or adapt IMGT software packages that are already existing. So the first step is the loci annotation. And for this, we have a current pipeline. So first is the extraction of the locus. So we extract the sequence from public database, and then we create a flat file with an IMGT accession number, and then we proceed to the locus annotation using internally developed tools based on IMGT ontology axioms and concepts. And then sequences are inserted into database. So we have LGMDB for sequences, GeneDB for genes, and 2 databases for 3D structure. Information about locus annotation are also on our web pages. And finally, reference sequence of ID and TR are inserted into IMGT tools for specialized alignment. So we have IMGT request, IMGT high request, and IMGT domain gap align. So first, have a better overview of what is available within IMGT. I've looked at our data. We have annotation for the 7 loci, but not for all strain, and data are not complete. First, I got interested into AGH locus to develop my methodology, and data are mainly available for C57BL6, which is our reference, and we have partial data for other strain. Because it was individual sequences that were annotated, we don't have complete locus, and here we have a table which summarizes the number of genes and alleles for each type of genes. So the first step of my PhD was to do an update of the annotation for the AGH locus for C57BL6J, and I did it on GRC M38P6 assembly. I annotated 238 genes, and here's the table of the number of genes for each types and their functionality, so functional, open reading frame, and pseudogenes. And compared to the previous annotation, I have 4 additional AGHV genes and 3 AGHD. Then I got interested in all the public data that were available, so I extracted AGH locus coming from 13 strains of the mice, and genomes are coming from the Mouse Genome Project. And I run an internal tools which is called LGM Motif that perform automatic annotation, and here we have the results for the 13 strain. So we have each type of genes, and as we can see, the number of genes for each type is pretty much the same, which means that we are more likely to have the whole locus, which is a good thing for my study. And then I compared those predicted genes to data that were already available on IMGT GeneDB. And this is all my preliminary results for AGHV genes. And I was looking for potential new genes, potential new alleles, and genes that were already identified in IMGT GeneDB in the same or in other strains. So in blue, we have genes that were already identified. In green, potential new genes, new alleles, sorry, and in purple, potential new genes. Besides C57BL6NG and LPG, it seems like the number of new alleles and new genes is higher than genes that were already identified. This shows that even if it's the same species, the AGH locus seems to present a high number of variation between strains. So I didn't finish yet the analysis, but this study will allow us to have a better knowledge of IG and TR repertoire of laboratory inbred mice, And this comparison will allow me to develop or adapting a methodology based on standardized IMGT gene description. And this will finally allow the enrichment of the IMGT database. So thanks for your attention, and thanks to the EMB committee for allowing me to do a presentation.
Thank you very much, Anna. And we have space for questions using your microphone or chat box, please.
So it's very interesting to look at inbred mice, and a lot of science is done on them. Even more, most of the data from gene ontology are based on mice and rat studies, which sometimes confuse us when we are trying to translate that to humans, because humans have a completely different metabolism. For instance, some genes are on or off. Just high, when you change the temperature from 24 to 25 degrees for mice. But for humans, that is not a very big issue that will never change. So it's very interesting to have even better catalog of the mice. But do you know if there are someone, or you are working in a way of translating all of this to humans? Because in humans, we do not have strains, but we still have some isolated populations that share some characteristics as the inbred mice.
Yes.
Any translator, easy way of doing?
I think that we are still working on haplotypes for humans, so data will be updated with a large number of data, because there are more and more sequencing of human. But the methodology is currently developed for mice, but it's aimed to be applied to other species as well.
Yeah, no, I do understand. I mean, I was just curious about if there was any kind of project that you know that is using what you have been producing, which is very nice, to a more translational aspect.
I don't think yet.
Okay. Don't worry, someone will start doing that, but it is so complex that it will take a long time. These are the first good steps to do.
Other questions from anyone? If not, I would like to thank again Anna